Alright everybody, we are continuing with Chapter 5, and thankfully as of this video, which I'm recording on Tuesday the 13th, I am finally starting to feel a little bit better now that I've been on a very strong anti-inflammatory medication for about 5 days now, but I'm able to breathe a little bit better now, thankfully, so hopefully I'll be able to knock out the rest of these videos here pretty quickly over the next couple of days. Alright, so we are talking about bone formation and development in this video. So we're going to talk a little bit more about the function of the cartilage that we find within our bone, and then we're going to talk about the two different types of bone development and formation, which are intramembranous ossification and endochondral ossification. And then we're going to talk about that growth activity at the epiphyseal plate that I mentioned in a previous video. And then we're going to look just a little bit about how bone is modeled, so initially formed, and then remodeling, which is repair of bone tissue after an injury like a fracture. Alright, so ossification also means the development or building of bone tissue. This is also sometimes called osteogenesis, so the creation of osseous tissue. So again, like we've mentioned before, bone does form off of a cartilage template, so that cartilage is going to give a framework for our bone cells to be building off of, kind of like if you see construction growing up and there's all the metal rebar that goes into it, that's kind of like our cartilage template, so it just gives us that basic structure on which to build out from. Alright, and so again, we have two different types of bone development: intramembranous ossification, which is how the flat bones of the face and the skull develop, most of the bones of the skull, or cranial bones, and then the clavicles are going to develop via intramembranous ossification. And then our other type is endochondral ossification, which is how the bones at the base of the skull and then all of our long bones in our body are going to be formed over time. Alright, so here's our diagram of intramembranous ossification. Alright, and so the way this works is that we are going to develop both compact and spongy bone tissue in this particular way, but the compact and spongy bone tissue is going to develop directly from that mesenchymal layer of cells that we talked about a couple weeks ago. So these are undifferentiated cells initially. All right, and so this is starting out as that cartilage template again. And so the mesenchymal cells in the embryo, so this is starting during embryonic development, all of those cells are going to kind of group up together. They recognize that they're similar cells that need to develop in a similar pathway, and so they will start to differentiate into their further levels of specialization. So like our osteoblasts and stuff like that are going to start to develop from those mesenchymal cells. And then some of those other cells will form tissues for the capillaries of the vessels that are going to run through all of this bone tissue that we can see here in these two lower pictures. So a lot of our structures here in intramembranous ossification are coming from those initial embryonic mesenchymal cells. All right, and so we start out with our osteoblasts here, so our bone building cells. So osteoblast, B is for build in this particular case. They're going to start out in something called an ossification center, which is shown here in image A. And so an ossification center, this is just a cluster of these osteoblasts in the early stages of this intramembranous ossification process. And so they are going to secrete something called osteoid, which is bone matrix that does not have calcium in it yet. So this isn't going to be super, super hard. So we see that osteoid tissue shown here. All right, so these kind of little dots around all of these osteoblast cells. So we're starting to get that matrix in there, that extracellular matrix. We're just not hardening it just yet at this point in time. It will start to calcify or harden over the course of a few days as we deposit those mineral salts, the hydroxyapatite, into it slowly over a few days' time, but we do not start out with very, very solid bones. All right, and so as that solidifies, it's going to trap those osteoblasts inside of that tissue, and they're going to kind of stay there, and then they're actually going to become osteocytes, which is what we're seeing here in panel B. So we've got that osteoid matrix has been solidified at this point in time. We've got that calcium mineralization there, so this is hard tissue, and so those osteoblasts are going to then further differentiate into those osteocytes, so kind of our housekeeper bone cells again, and so that kind of traps those osteocytes in there. And then to keep getting osteoblasts, because we do still need to be developing new bone tissue, those osteoblasts stay in this ring around the outside of that internal matrix where we have started solidifying that particular tissue. All right, and again, this is all being built on that cartilage framework. Okay, so that kind of gives us that little bit of a building block on which to develop some of this harder tissue. Just gives us that framework. All right, so we've got our osteoid, our solidified osteoid here. We've got osteoblasts ringing that so that they can keep building bone tissue outwards. And then we've got those housekeeper osteocytes inside to help maintain that bone tissue deeper inside of the whatever bone that we're looking at. All right, and so we see this again in both compact and spongy bone, so it's going to look just a little bit different depending on what type of tissue that we're developing, but these upper two pictures are mostly representing the compact bone development, but it's going to be a very similar process for spongy bone, which we see diagrammed further down here in panels C and D of these images. But same idea, we are going to go ahead and secrete that osteoid matrix into this framework, the trabeculae that we talked about in a previous video, and then all of these blood vessels are going to be intertwined throughout that network. So again, this is a sponge-like tissue here. But if you were to kind of chop one of these trabeculae pieces here kind of in half, like right about this way, and look at a single end of it, it's going to look very similar to panel B up here, because we are building that bone tissue out in this framework here. All right. Those mesenchymal cells can also differentiate into fibroblasts, which are what's going to form that fibrous periosteum on the outside surface of our bone tissue, which is what the tendons and ligaments are going to attach to again. All right, so that's also coming from those mesenchymal cells. All right. Let's talk about endochondral ossification now and how these two processes are a little bit different. All right, so again, remember that intramembranous ossification starts during embryonic development. So this is starting during the embryonic stage before somebody is born. It's also referred to as being in utero. Right? This will continue after being born well into adolescence, so kind of the late teen into early 20s, that intramembranous ossification process will continue, but it will kind of drop off as we get into adulthood. All right. Endochondral ossification. So instead of using that cartilage as just kind of a base framework to build off of in endochondral ossification, we are actually going to replace hyaline cartilage that is present in these developing bones. All right, so intramembranous ossification is using that cartilage as kind of a template, a framework to build out from, but it is not replacing that cartilage. whereas endochondral ossification is going to be replacing those cartilage plates on the ends of bones. All right, and this has a little bit to do with what specific bones develop this way. So if you remember intramembranous ossification, we're looking mostly at bones of the skull and then the clavicles that are being developed through intramembranous ossification. And our longer bones are going to be endochondral ossification. And part of that reason is, again, that intramembranous ossification, this is a slower process. We've still got some of that cartilage in there for some flexibility, and those bones are not solidifying immediately upon starting to develop. Remember, let's go back to our previous slide. Remember, this osteoid matrix is not going to be solidified initially. And so that's really beneficial during birth, actually, because the skull and clavicles are going to need to be able to kind of deform a little bit to pass through the birth canal. And so if we did this endochondral ossification where we're replacing that cartilage and losing that flexibility, then those bones would not be able to deform a little bit in order to pass through the birth canal, and that would make it very, very difficult, if not impossible, for that baby to be born the way that it's programmed to. This is where things like a C-section would come into play, needing to have a baby come out through C-section. But that intramembranous ossification is going to help make sure that the bones of the skull and the clavicles can bend a little bit during that birthing process so that it's easier to pass through the birthing canal. And then as the child grows, then those bones are going to finish solidifying and actually become hard, and then the bones of the skull will fuse together and stuff like that. All right, so endochondral ossification, again, we are replacing that hyaline cartilage instead of just using it as kind of our base framework. So the cartilage itself is not going to like turn into bone specifically, so the cells that are in that cartilage are not going to differentiate into bone cells. They do not have that ability, because once they're those cartilage cells, then that's as far as they can go in that differentiation process. So they're not going to turn from a connective tissue cartilage cell into a bone cell. That's not going to happen. All right, but the cartilage gives a template that is slowly broken down by the bone cells as they are building that bony tissue. All right, so it's still using it as a framework, but it's also destroying the cartilage in the process. Okay. So endochondral ossification is going to take much, much longer than intramembranous ossification does. So the bones at the base of the skull, so our occipital bone, which we'll talk about in our next lab unit after this exam this week. So the occipital bone and the temporal bones are going to be formed this way. And then again, our long bones, which are a lot of the bones in our body, the femur, tibia, fibula, humerus, radius, ulna, etc., are all going to be formed through this process. So what we have diagrammed here in this slide is the development of a long bone during endochondral ossification. So these can start developing at about 6 to 8 weeks post-conception. So we're still going to get differentiation from mesenchymal cells, but those are going to differentiate into chondrocytes, so those cartilage cells that are going to form that cartilage skeleton. So that's what we see up here in panel A. So right here, this is just cartilage. There is no bone tissue in panel A right here. It's just that hyaline cartilage. All right. And so that is also going to have something called the perichondrium on it. So that's a membrane that's going to cover the cartilage to kind of keep all of these cells and other tissues encased and keep them all together near each other. All right. And so as we produce more and more of this matrix slowly over time, through these chondrocytes and some of these bone cells that are going to start to appear in this particular tissue, we're going to start that ossification process, all right? So that matrix starts out uncalcified, just like we saw previously, and it will slowly calcify over time. All right. Ooh. Did not mean to do that. I'm sorry. So that matrix that we are using that cartilage as a basis for, we're slowly going to start adding that calcium mineral to it, and it's going to start hardening that tissue slowly over time, which means that the nutrients that were previously getting into those chondrocytes, the cartilage cells, they're not going to be able to get those nutrients anymore. All right, because we're starting to solidify in here, and that's going to prevent that nutrient movement, which means that those chondrocytes are going to die because cells are not going to survive if they are not getting the nutrients they need. All right, and the death of those chondrocytes is going to lead to the rest of that cartilage starting to disintegrate, because if we don't have cells to maintain it, then it's going to break down slowly over time. And then we're going to start getting blood vessels starting to spread throughout this calcified matrix. So before that calcified matrix gets super solid and really packed in there, we want those blood vessels to start coming in there and passing through, so that they're not getting blocked out further in that particular development process. All right. And these blood vessels coming in is also going to help start making this cavity in the diaphysis region of these long bones a little bit bigger. So it's going to help start expanding these bones a little bit, because they're going to take up space too. And they are going to start bringing a lot of these osteogenic cells with them. So those stem cells for bone tissue, those are going to develop into our osteoblasts. All right. And so this initial part here, this is eventually going to become that medullary cavity surrounded by that compact bone, and then we'll have our spongy bone tissue out on the proximal and distal ends of those bones. All right, so we got to get those blood vessels in there to start bringing nutrients and our osteogenic, our bone stem cells, so that we can differentiate those into our osteoblasts, which will further start developing and building all of this bone tissue. So panels A, B, and C here, we're not really seeing bone cells in here just yet. We're starting to get those calcium deposits in the matrix, but we're not getting actual bone tissue being built just yet. That transition kind of happens here in between C and D. So as those arteries are coming in, bringing in those osteogenic stem cells, so that we can start actually building that bone tissue. All right. And so the vessels and the new cells that are being brought in, as well as the continued calcification of that matrix, is going to start pushing the two ends of this cartilage template away from each other, which is what's giving those bones their length. And then obviously we're going to build out in width or diameter a little bit as well, but we're really focusing a lot on length with these bones. We need to get them longer. All right. This is going to slowly over time start depositing more and more of that mineralization. So we get that compact bone on the outside of that medullary cavity. That perichondrium that we saw earlier is going to be able to help us develop that periosteum that we're attaching our tendons and ligaments to. So that does have a function other than just keeping that cartilage contained. It's going to give us a building block for that periosteum layer. And so we start calcifying that medullary cavity and that compact bone in the diaphysis region first, leaving some of this cartilage up here at the ends. All right, so we've still got a lot of cartilage up here that's going to be at the proximal and distal ends of these bones. and then we will start calcifying those ends. All right, so in panel D, as you can see, the end of that bone is still uncalcified matrix, and the calcified matrix is down here, more in that metaphysis region of the bone rather than the diaphysis. And then our medullary cavity is pretty much fully formed here with that diaphysis down here. So that's pretty well established at this point in time. These vessels are going to keep spreading throughout the bone, and then over time we are going to get additional arteries and veins coming into the proximal and distal ends of the bone, bringing in more of those bone cells, so our stem cells for building bone. And this is going to create a secondary ossification center. So our primary one is down here in the medullary cavity. That's what forms that medullary cavity in there. And then our secondary ossification centers are going to be at the proximal and distal ends of the bone, and that's what's going to form that spongy bone tissue in those regions. All right, and so that will start hardening that matrix again. So we'll start putting our calcium deposits in there slowly over time, and as you can see, that's going to start kicking out all of that cartilage tissue. So again, as we start to calcify that matrix, the chondrocytes are going to die, which means they cannot maintain and rebuild that cartilage tissue. So it's going to slowly disintegrate over time, allowing it to be replaced by those actual bone cells and the subsequent bone tissue. All right, and so by panel F down here, we've got just a little bit of cartilage left here at the ends. This is going to be on both proximal and distal ends, but we are left with this articular cartilage that's going to stay out on the surface of those bones. That's going to stick around long term because that's going to help provide some cushioning and reduction in friction for our joints. So we want that articular cartilage. And then we're going to keep a little layer here in between where we're going to have the spongy bone and the compact bone and medullary cavity, and that's going to be that epiphyseal plate or epiphyseal line that I've mentioned in a previous video. This is also sometimes called the growth plate, all right, because this is still going to be cartilage that we are able to keep building off of in length for these bones. All right, so again, endochondral ossification, this is going to take a very long period of time. And so most of the long bones in our body, especially like the humerus and femur and like those bigger bones in our arms and legs, that epiphyseal plate is going to be around until usually adolescence. So again, late teens to early 20s is when we finally see that epiphyseal plate fully disappear, because that's how long it actually takes to build out on that cartilage template and grow these bones in length. But if we built that too early and we solidified that too quickly, then the bones are not going to grow in length anymore. All right. And so the exact point in time that this stops and how quickly it happens varies from person to person because our genetics are different. Everybody's a little bit different height, right? So those differences is kind of how we get some of these height changes, all right? So let's look a little bit closer at that epiphyseal plate. So that's kind of what we're seeing here in this particular image, is we're looking closer at that epiphyseal plate or epiphyseal line. All right. So we're going to have, again, that layer of hyaline cartilage. So that's up here. We've got some changes happening in those chondrocytes. They're going to start, again, dying off. And these vessels are going to start bringing our bone cells, and we're going to have proliferation, so growth and replication of those bone cells right here. They are going to mature and develop into those full osteoblasts, and then further on from there as needed, and that's where we're going to start that calcification process and giving us that zone of ossification. All right. So we are basically just kind of slowly building out from that line of cartilage. giving us longer and longer and longer bones slowly over time. All right. But remember, those chondrocytes up here, they're not helping with bone growth. These cells are just going to die off. They're not going to turn into actual bone cells. All right, let's talk a little bit about how bones grow in diameter now. So how do they get bigger around? Right, so this is going to happen simultaneously as the bones grow in length from that epiphyseal plate, but we do still need to get our bones to be bigger around, because it doesn't make sense to have a super long bone that's also really skinny. So if you think about like a tree branch, the smaller in diameter that branch is, even if it's super long, it's still going to be really fragile, versus a branch that is much larger in diameter, that's going to be a lot stronger. So similar idea with our bones, we want that strength. We don't want to be risking breaking our bones super easily. All right, so this is happening simultaneously, and the growth in diameter is called appositional growth. And so what happens here is osteoclasts, so the cells that are breaking down bone tissue, they are going to reabsorb older bone inside of that medullary cavity. So they're going to slowly break that down a little bit, not super quickly, it is going to be pretty slowly. And the osteoblasts, so building bone cells, they are going to produce more bone tissue right underneath that periosteum, that outer covering. All right, so we are simultaneously breaking down the oldest cells in here and developing new ones and new bone tissue further out. So that's how we get that growth in diameter that makes our bones larger around and makes them a little bit more durable and stronger in the long term. All right. This process is called modeling. All right. So this is that reabsorption on one surface and building new bone on another surface is modeling. So that's just normal growth, is bone modeling. And in the next video, we will talk about bone remodeling or fracture repair.